Hey everyone, welcome back to a new chapter, and in this chapter we are going to start looking at some specifics surrounding how genetic information is eventually used to synthesize proteins. So we're going to be talking about not only the genetic code, but also the transcription and translation processes. In this first video, we're going to be focusing on the actual genetic code, but first we're going to look at an overview of the entire process, which we call the Central Dogma of molecular biology. So the Central Dogma of molecular biology really pieces together a lot of important experimental breakthroughs that we've kind of been talking about over the past few chapters. This includes not only Gregor Mendel's work, but everything leading up to the current day, including the work of people like Meselson and Stahl and Hershey and Chase. And this puts it together in a model of how genetic information is eventually expressed into physical traits. Now, we've talked before about DNA replication back in chapter 14, and of course we know this is necessary for cell division to occur, whether it's mitosis or meiosis. There's always a S phase in which DNA has to be faithfully replicated so that each daughter cell has an equivalent amount of DNA. So that is one part of the central dogma in the sense that DNA is propagated through the DNA replication process, which is catalyzed by DNA polymerase. Everything else you see below the DNA replication makes up what we consider to be protein synthesis. Genes on the chromosome, and you'll recall that a gene is just a segment of DNA that provides the instructions for making one protein. Genes on the chromosome are transcribed into a messenger RNA molecule. This messenger RNA is just a copy of the gene in RNA form that can then leave the nucleus and then can go out into either the cytoplasm or rough endoplasmic reticulum and be translated into a protein by the ribosome. So as we've seen, the messenger RNA is going to serve as the instructions that the ribosome will use to synthesize a protein, and the messenger RNA includes the specific details, including how many amino acids are we using, which amino acids are we using, and in what order do those amino acids go when we're building the protein. So once we have all of our proteins made, the proteins that our cells actually express are therefore going to be responsible for our physical characteristics, which of course we know as our phenotypes. Just a quick overview of the transcription process. We're going to have later videos that delve into this topic in a little bit more detail. You'll recall that both DNA and RNA use a language that only contains 4 letters, and these letters come in the form of nucleotide bases. DNA uses A, T, C, and G, whereas RNA uses A, U, C, and G. So the difference is whether or not DNA or RNA use T or U. So transcription is going to involve the unwinding of DNA, very similar to what we saw with DNA replication, and then the synthesis of a complementary RNA molecule on top of one of the 2 template DNA strands. So in this particular case, this bottom strand here that is read from right to left, 5' to 3', or 3' to 5' left to right, this is what we would call the template strand. The other strand on top, the one that goes from left to right, 5' to 3', that is the non-template strand, but it can serve as a template, just not in this particular case. So the RNA molecule is being synthesized, and you will notice that the RNA that we are synthesizing is complementary to the template strand down on the bottom. So the A goes to T, the U goes to A, the G goes to C, and so on and so forth. Now, the enzyme that is actually synthesizing this RNA molecule that is complementary to the template strand is called RNA polymerase. Not DNA polymerase, but RNA polymerase. So it's a little different than what we saw with DNA replication. But something that is similar is the fact that this synthesis goes always from 5' to 3'. So the first nucleotide that was incorporated was this A right here, and then the newest nucleotides that are being incorporated are on the 3' end. So 5' to 3'. So again, we'll talk more about this later on, but after an RNA molecule is synthesized in the transcription process, next that RNA needs to be processed and edited, which is something we really haven't talked about a whole lot. So before the mRNA leaves the nucleus, it needs to be processed because it is not quite in a mature form and it is not quite ready to be translated into protein. So A immature RNA molecule contains two types of sequences. Exons are segments of the mRNA that contain vital coding information and must be preserved. You might think of them as containing the absolutely essential information for building the protein, but these pieces of information are not necessarily always right next to each other. Introns are non-essential segments of essentially nonsensical information that are in the RNA that actually need to be cut out before the RNA can be translated into protein. So think of it like this. Say you have a book that you're reading, and every 5 pages you have a page that just contains a whole bunch of scrambled code that doesn't mean anything. In order for the narrative in the story to make sense, you would need to rip those pages out. That is essentially what the cell has to do. The cell, through RNA splicing, uses enzymes to remove the introns and then stick the exons together so that they are contiguous pieces of information that are all read in one go. So nuclear enzymes in the nucleus need to splice these exons back together after the introns are removed. And then once that is done, we actually cap both the 5' and 3' end of the RNA. So we'll talk more about that later, what these purple things are here and what this long string of A's is over here. This essentially serves to protect the stability of the RNA. And at this point, the RNA we would consider to be mature, and it is now ready to leave the nucleus through an available nuclear pore, and it will go out into the cytoplasm to bind to a ribosome, which can then translate it into protein. Okay, and now just a very quick overview of the translation process, and again, we'll delve into this in more detail later. We've seen before, and we've talked about this in some of the previous kind of seminal experiments in molecular biology back in the 1800s. But one of the challenges is that DNA and RNA, yeah, they both use a 4-letter code. Proteins use a 20-letter code, though. You'll recall that proteins are made out of 20 different types of amino acids. So we've covered this challenge before, but the idea is that if mRNA were decoded one base at a time, then we could only code for up to 4 different amino acids. An A would correspond to one kind of amino acid, a U would correspond to another kind of amino acid. So if we literally only read the mRNA or the DNA one nucleotide at a time, we could not possibly code enough information to cover all 20 amino acids. So what we do instead is the ribosome will actually read the messenger RNA in groups of 3. These 3 bases are called codons. And when you read nucleic acids 3 bases at a time, this actually provides up to 64 different possible codon combinations, and that ends up being well more than enough to code for all 20 amino acids. In fact, that is so much more that we actually have a lot of redundancies in the genetic code, which we will see on the next few slides. So as the ribosome scans through the messenger RNA, transfer RNA molecules come in bringing and delivering the appropriate amino acids, which correspond to the codon that is found in the messenger RNA, and the ribosome will use its enzymatic activity to build peptide bonds between the amino acids as they are brought in. Again, this is an oversimplification of what happens during translation. This is just an overview. We'll talk more about it in greater detail in a later video. So as we start talking about the genetic code, let's revisit the nature of all 20 of our amino acids. The 20 amino acids, depending on kind of how you like to classify them, we can put them into 5 different classes based on the chemical properties of their side chains. You, of course, have the hydrophobic, nonpolar amino acids like glycine, alanine, and valine. You have the polar amino acids like serine, threonine, and glutamine. You have positively charged acidic amino acids like lysine and arginine. You have acidic negatively charged amino acids like aspartate and glutamate. And then you have aromatic hydrophobic ones like phenylalanine and tyrosine and tryptophan. So obviously, these chemical properties of the side chains are very important because it's going to be a major determinant of how the protein eventually folds. And we're going to find that protein folding actually happens very soon after the protein synthesis process gets going. So, because the way the protein will actually end up folding is going to very much be dependent on the primary sequence of amino acids within the protein, it's going to be very important that the messenger RNA be read properly so that we put the protein together in the correct way. We want to make sure that we're putting all the amino acids together in the proper order. If we make any mistakes in that, the protein is going to fold differently and it is going to function very differently too, and that may end up causing the cell a number of problems. Okay, so do you remember how I said that the genetic code includes a lot of redundancies? Well, here's what we're talking about there. Sometimes we use the term degenerate when we're talking about the genetic code, and that doesn't mean that the genetic code is like some sort of menace to society or anything like that. No, the word degenerate here means that the same amino acid is often coded for by more than one kind of codon. So again, redundancies. You'll notice that for threonine, there are 4 different codons that code for that amino acid. For leucine, there are 6 different codons that code for that amino acid. For arginine, there are 4 of them. For glutamine, there are 2 of them. So it's really kind of a rare occurrence where there's only one codon for one amino acid, methionine being a prime example of that. So real quick, you may want to pause the video after I read this question. Look at the codon table to the right. So this codon table is telling you for any particular codon, so on the left here that would be the first base, the second base on top, and then the third base on the right. For any particular codon, what do you notice about the codons that code for the same amino acid? So you might look at the ones that code for proline or the ones that code for serine. So pay attention to the first, second, and third bases. What do you notice about the codons that code for all the same amino acid? You might want to pause the video and then we'll reveal the answer when you come back. One of the things you'll notice is that, say for proline, the thing that is the same is that the first 2 bases are the same. So proline always requires that your first 2 bases be C. Serine requires that your first 2 bases be U, then C. Threonine requires that your first 2 bases be A, then C. It turns out that in a lot of cases, the third base is not really all that important. The third base is sometimes inconsequential because of what we call wobble during the translation process. That third base is sometimes not always read correctly, so to counteract this problem, the genetic code has evolved so that the third base doesn't matter all that much. So if the third base doesn't get read correctly, then you'll still insert the same amino acid regardless. So this third base makes it more likely that a random mutation will likely not have a huge impact on the amino acid sequence of a protein. And you'll recall that we call those types of mutations silent mutations. Okay, so a couple of important codons to take note of here, and we've talked about these things before. Every reading frame will always begin with the start codon, which is AUG. So we'll kind of cover more by what we mean by reading frame in the next few slides. But the start of every protein has to be AUG. The ribosome does not start building a protein until we encounter that start codon AUG. So the codon always codes for this amino acid methionine, which explains why most proteins actually start with methionine. The only proteins that do not start with methionine actually probably had the first part of the protein chopped off. So once the ribosome encounters the start codon in an mRNA transcript, this establishes the reading frame. This sets the reading frame and tells the ribosome, this is where you start building your protein. You don't start building the protein 1 base before or 1 base after. You start building it right here. And then, of course, we have our 3 stop codons, UAA, UAG, and UGA. They mark the end of the reading frame and they signal the ribosome to stop adding amino acids to the growing protein because we are done. You don't need to add any more amino acids. This is the end. Okay, so let's talk a little bit about what we meant by the term reading frame. So interpreting a messenger RNA transcript can sometimes be like decoding a complex message. Often you need some kind of decoder ring in order to figure out where the message is. You have to be told where to look for the message. So to give you the idea of what we're talking about here, consider the following messenger RNA sequence. From 5' to 3', it goes UUCAC GGA UG CCC UAC GCA AGC 4As CGU GAC UAA. Okay, so what we can see here is that there are three different ways to interpret this message. The first way, the first reading frame that we can see here, is to read 3 codons at a time, starting with this U right here. So the codons that we would be reading are UUC, then ACG, then GAU, then GCC, and so on. So whatever we decide is our reading frame is going to set which amino acids get incorporated into the protein. But we're going to see there are actually 2 other ways that we can interpret this message. But if this is what we choose as our reading frame, ignoring the fact that we need a start codon, then the reading frame here would be phenylalanine, then threonine, then aspartate, then alanine, leucine, arginine, lysine, glutamine, asparagine, valine, threonine, and so on and so forth. Again, ignoring the fact that we ordinarily would need a start codon. So that is one reading frame. Let's look at a different reading frame. What if instead of reading in triplets UUC, ACG, what if instead this UCA right here is what we choose to read as our codon? If that starts the reading frame, then suddenly our reading frame becomes UCA, then CGG, then AUG, then CCC, then UAC, and so on. If that's the case, then our reading frame would look completely different. UCA codes for serine instead of phenylalanine, so this reading frame would go serine, arginine, methionine. You'll notice that that is actually supposed to be the start of the reading frame because that's your start codon. Proline, tyrosine, arginine, serine, lysine, threonine, and then finally a stop codon. So you can see that UGA there is a stop codon, so we have to stop right there. And finally, we actually have a third reading frame. Instead of going UUC as your first codon or UCA as your first codon, you could actually go CAC as your first codon. If you set the reading frame that way, you once again get a completely different amino acid sequence. In this case, you get histidine, glycine, cysteine, proline, threonine, glutamine, alanine, lysine, arginine, aspartate, and then finally you hit the UAA stop codon at the end. Again, this ignores the fact that you ordinarily need to start with a start codon, and the only reading frame that we can actually see here that includes a start codon is the second reading frame. So the reason why I bring this up is because every messenger RNA transcript has 3 potential reading frames, but only one of those reading frames is actually correct and includes the correct message and the correct instructions for building the protein. The way that we ensure that we always pick the correct reading frame is by making it such that the protein synthesis does not start until you encounter the start codon. The start codon sets the reading frame and ensures that the correct information gets translated into protein. Now, one type of mutation that we really have not talked about before is called a frameshift mutation. A frameshift mutation includes either the addition or deletion of 1 or 2 nucleotides, and what this does is it shifts the reading frame from the appropriate one to one of the 2 inappropriate ones. So looky here. If the correct reading frame is supposed to go AGC, which corresponds to serine, GUA, which corresponds to valine, CCC, which corresponds to proline, and UAC, which corresponds to tyrosine, if we accidentally delete those 2 nucleotides right there, that shifts the reading frame. So now instead of serine, valine, proline, tyrosine, it's serine, alanine, leucine, leucine. So by shifting the reading frame, we have entirely changed the message that happens after the mutation occurs. And we are going to entirely synthesize our protein in a completely different way, and that can cause some really, really, really big problems. So anytime you have a mutation that involves the addition or deletion of nucleotides, you really, really want that deletion or addition to be in a multiple of 3, because if it's a multiple of 3 rather than a multiple of 1 or 2, then you do not end up shifting the reading frame. You might add an amino acid or lose an amino acid, but at least you don't shift the reading frame. So frameshift mutations are right up there with nonsense mutations in terms of being the absolute worst kinds of mutations you can have. So like I said, these types of mutations tend to be really devastating, since the protein will almost certainly be made in an entirely different way because you've shifted over to one of those inappropriate reading frames, and you may even end up with a protein that's either too short or too long, depending on where that frame shift leads into a stop codon being reached. Okay, so we can kind of tie all this together, and we can actually tie this into some of the evolution stuff that we've been talking about. So one of the major pieces of evidence that suggests that all organisms on Earth have a shared ancestor and a common ancestor is the fact that all cells contain DNA, and with very few exceptions, the genetic code that we talked about is universal. Bacteria, human cells, dog cells, fungi cells, plant cells, we all use the same genetic code. That codon table that we saw where a codon corresponds to an amino acid, almost every kind of cell uses that exact same method for synthesizing proteins. So that suggests that we all have a common ancestor because we all deal with our DNA information and our protein information in exactly the same way. A good example of this being in action is in the biotech industry. Human genes like insulin, so insulin is a big biotech industry because diabetics always need synthetic insulin that they can inject themselves with to control their blood sugar. Human genes like insulin can be spliced out and then ligated into bacterial vectors like this circular piece of DNA that you see here. And then the bacteria can be transformed with this recombinant DNA so this is a piece of bacterial DNA that actually has a human gene spliced into it. So because of the universality of the genetic code, the transformed bacteria will actually make insulin in exactly the same way a human cell would have. That would not be possible if the genetic code were different between bacteria and human cells. All right, that's going to do it for this first video. I hope you enjoyed it. So that's going to be a good jumping off point for our next discussions on transcription for the next 2 videos and then translation for the last few videos. Thanks again, and I will see you next time.